yesterday, if you were here, there was a lot of discussion about the cell repair or the damage repair hypothesis, which Aubrey has been a big proponent of, and its validity and its implications. Our goal this morning is to take that beyond just the validity of the concept and of the hypothesis, and to think about how we might translate those insights into real cures, or at least real treatments. So we got 4 very interesting people to talk about that. First off will be Daniel Kraft, who has had a very interesting and himself diverse career. I noted from his bio that he's interested in convergent and exponentially developing technologies. The mathematics of that rather defeated me, but he's going to talk about the future, where he sees the future in these fields going. He's an entrepreneur as well as a futurologist, so he's going to help us with thinking about how businesses might be created, which was a theme of the previous session. He wants me to give his apologies that he may have to slip out before the end of this. May have to, depends on how it all goes, because he's got double booked somehow. I don't know how that works, but if you're a futurologist, you can't predict the future, right? I think that's how it works. Okay, so after that, we have Judith Campisi from the Buck Institute on Aging, has done a lot of very leading edge work on cell senescence, has had a very interesting career that embraces Harvard Medical School, BU, Lawrence Livermore Laboratory, as well as the Buck Institute. So she's going to tell us a little bit about how cell senescence might lead us into the future of dealing with aging. Then we'll have Julie Allickson, who is very much embedded in the whole translational research agenda in relation to regenerative medicine. So she's had a very interesting career, and both academic and company, and is now, as I say, head of translational research for Wake Forest. And then finally we have Howard Foyt, who was telling me he started work in academia. I'm not sure it's all fully in his bio. Then he went to Park Davis. Then he was bought, along with many other people, by Pfizer, but then has subsequently worked at 2 or 3 biotech companies, and now in this very fascinating position working at ViaCyte. So I just want to say one thing, which is like a personal anecdote before we get going with Daniel. I was sitting on a plane just a week ago with a woman who is actually a high-flying academic who clearly had very advanced rheumatoid arthritis. You could see that in her hands and her movements. But she had recently developed interstitial lung disease. And so that sent me to the web, and turns out that about 30% of people with rheumatoid arthritis also have some kind of detectable lung problems, which then takes you back to, what is happening in the immune system in both cases. In one case leads to arthritis, in another case leads to lung disease, and what's causing that? What's the process that's underlying that? And so one of the themes of this session will also be, instead of just looking at the diseases that present at the top of the pyramid, what are the underlying biologies at the root of it? So anyway, that's all I wanted to say, and over, Daniel, to your presentation.
Thanks. So maybe I'll stand up. I'm just going to talk for maybe 5 or 6 minutes and help, if I can pull my slides up, a little bit of maybe framing about the future of regenerative medicine and beyond. I trained in internal medicine, pediatrics, and hem-onc and bone marrow transplant at Stanford, and spent a lot of time in the lab as well. And I think it's really interesting as we are in this new age about rethinking not just how we think about aging and rejuvenation, but think about how we can apply many of these really fast-moving technologies into this field and into research in general. I've had an interesting seat to look at convergence and exponentials, sharing the medicine track just up the road at Singularity University. We have tracks also in biotechnology, robotics, artificial intelligence, 3D printing, and beyond. And it's been really interesting to bring very bright folks together to look at how we can apply fast-moving technologies to address grand challenges. And I think we have several alumni and faculty here. Aubrey's been on faculty several times. And part of the theme partly driven by our co-founder, Ray Kurzweil, who's now director of engineering at Google, is to start thinking exponentially and realizing, like with Moore's law, our smartphones have about 1 billion times the price and speed of performance of the best supercomputers in the 70s. Your iPads are better than a crazy supercomputer in the late 80s. Where's technology going on these exponentials, which is often hard for us to sort of conceive of that, what will the iPhone 10 look like, for example, and how could we leverage that into research, into aging and connected and digital health? And realizing, again, it's not just Moore's law that's getting faster, it's and cheaper and better. It's many of these fields at their convergence point, which I think enables us to rethink how we do research, how we take care of ourselves, aging and beyond. At Singularity University, we think a lot about how to address grand challenges. Many of those are in healthcare. We had our graduation of our 6th summer class last night, and about half of the projects were actually biomedical related. Some very interesting things I think you'll see coming out of that realm, from 3D printers, which will be flying to space station in the fall, to others that are doing home-based sensors to detect cancer and beyond. And as part of my role there, I put together a program called FutureMed, which focuses on the convergence of technology and reinventing healthcare. Aubrey will be back on our faculty this fall. We've grown the program to be a bit larger, now called Exponential Medicine, being held in the fall at the Hotel Del. So please join us there. So one of the things I want to help reframe here is that we often think in our own buckets. I trained in hem-onc, or I spent time studying hematopoietic stem cells. But I think where we often lack is the overlaps, and part of I think the theme here is that it's this overlap of fields that I think enables us to put things together in smart ways, whether it's crowdsourcing biology and research with groups like Foldit, where there's been a lot of interesting impact in crowdsourcing research. There's new platforms for bringing sort of social networks of physicians or clinicians and researchers together that have been quite impactful. It was published in Nature a week or so ago about the impact of putting together research in new realms. We're seeing companies being formed. One upstairs from us at Singularity University called Scanadu has basically a medical tricorder coming out. We'll start clinical trials this fall as part of an X Prize competition. And sort of the integration of all these things, whether it's thinking about longevity or prevention or better diagnosis, is I think how we're going to start bringing in the citizen scientists, the crowdsourcing of research, all these omics information, and putting them together and blending them in smart ways. And I think we can start really to apply that to the field of rejuvenation and regenerative medicine. This is my favorite chart of the hype cycle, which often really happened in the stem cell world. You remember the dearth of disillusionment, and we're entering the realm of enlightenment, and many of the technologies and the folks in this room are really starting to bring these elements to the field, into reality, into actual clinical applications. But there's also the sort of the low-hanging fruit. I started my clinical life as a bone marrow transplant physician. We have lots of sources of adult stem cells today, and I think like anything else, it comes down to your unmet need. When I was a bone marrow transplant fellow at Stanford, I spent a lot of time in the lab, or in the OR, harvesting bone marrow the old-fashioned way, and as part of the biodesign program, ended up coming up with a new approach to harvest marrow in a minimally invasive way. And so I think when you bring together engineers, device folks, we can think of ways. In this case, we have a technology I'll sort of skip through, but we've now got FDA approval and the devices through clinical trials to more rapidly harvest adult stem cells for my field of bone marrow transplant, but now potentially to be applied in many of these other realms of regenerative medicine, where with a single puncture you can do an entire harvest in 10 minutes and maybe bank those cells for future regenerative use, since there's some interesting applications of that as well. And we're now in the phase of building out the next device. So obviously with the realm of iPS cells and the sort of democratization of kits where almost anybody can create their own stem cell lines in their labs, even from themselves, there's a lot of interesting opportunities to blend that with other fields. We'll hear more from Wake Forest, I think, about my stuck slide about regenerative. Can you hit the next slide? About blending 3D printing and beyond. Oh, we're stuck. Technology fail. We have a saying at Singularity University, AI is easy, artificial intelligence, but AV is hard. So maybe we'll just leave it with this slide, and I'll close by saying, it's the ability now to democratize research, to do some pretty incredible things, building our own individual iPS cells, blending that with things like 3D printing, synthetic biology, and shifting healthcare in terms of longevity side from something that's traditionally been very reactive and intermittent. You wait for the heart attack, the stroke, the cancer, the aging to happen, to a field which is much more continuous and proactive. We'll have the Internet of Body. I'm wearing 3 or 4 sensors right now. We'll have home-based tricorders. We'll have our omics and other things crowdsourced. We'll have our own sort of check engine light for the body, so we'll understand before we hit the wall or have an acute or chronic event develop that we can have intervention. And I think it's the blending of the sensibilities, both in the rejuvenation and stem cell world, and also in how we rethink how we do research across the world. Bringing on the next billion minds, which will be online in the next few years, to reboot and rethink health, medicine, biotechnology, and research. So with that, I'll say thanks.
Judith.
Can you hear me? Yep, okay. So I'm not going to give you the 30,000-foot view. I'm not going to give you the 300,000 view. I'll try to give you something in the middle, which is really the challenge, I think, that we face in both extending lifespan but also extending health span, is trying to understand what it is that is driving the major things that actually kill us. So nobody dies of good health. You die of disease. And we know that there's a panoply of diseases that are driven by something we call aging. And the big challenge is to really understand what that is. So I think one of the things that Aubrey has done is put forth the idea that this is really damage, accumulated damage. And I want to put this in a slightly different context, which is that damage is rarely the problem. And that's because throughout evolution, all organisms have evolved really, really good ways of dealing with damage. They had to. This was survival. The problem is repairing the damage is never 100% efficient, so there are mistakes that are made, billions and billions of damages to macromolecules every day. Repair systems don't always get it right. And this is, of course, most serious for DNA. This is the one molecule in our cells that doesn't undergo constant turnover. So that's one problem. The other problem then is if the damage is a little bit overwhelming, cells will change their phenotype. And for the purposes of driving aging, the two main phenotypes are cell death and this process called cell senescence, which is a little bit more complicated and involves shutting down proliferation, but also developing a phenotype that tells the surrounding tissue there's a problem. So that all sounds good, and it is good. It is undoubtedly what keeps us healthy and fit for about half of our lifespan. But after 5 or 6 decades in the case of a human, what am I doing? 5 or 6 decades in the case of a human, a year and a half in the case of a mouse, these decisions of repairing not necessarily 100% correctly, or opting to die, a cell opting to die, or opting to allow the tissue to know that there's a problem, begins to be what we call maladaptive. So I think the real challenge is really identifying which of these options of responding to damage, repair, dying, or undergoing senescence, is the major driver behind the diseases of aging. And then, of course, we're never going to be able, at least I think in my lifetime, what's left of my lifetime, to devise a drug that's going to tackle aging per se, but try to tackle a disease based on this underlying problem that we see. So repair. We, meaning basic biologists, we do not yet know of a single way to effectively improve DNA repair in any cell type. So there are probably a dozen different DNA repair mechanisms, and the problem is they all talk to each other, and you can't just simply imagine overexpressing one component or another component, because these are multi-protein complexes, and as soon as you overexpress one, the others start not to function very well because of all these interactions. So this is really a challenge, and I personally have not seen a good idea of how to improve repair. So the next way to tackle the problem is to think about the two cell fates. So one is apoptosis. And there, I think stem cell research has great promise, because cells are dying, and we do have, at least in principle, therapies that we think can replace those cells that have undergone cell death. The last cell fate, cell senescence, I think is also a very formidable problem, although at least I think there are some ideas out there about what to do about it. The problem with the senescent cell is it doesn't die. And so it sits there and it's communicating with its neighbors and telling the tissue there's a problem, and eventually that tissue becomes inflamed, and we all know inflammation drives many of those diseases that will end our lives. And so the initial idea was to try to identify those pathways that drive senescent cells to communicate with its neighbors and interrupt them. And I think that paradigm is still valid, but I think there's a better paradigm, which is try to get them to die. Apoptosis is actually a great way to eliminate a damaged cell because there's usually no inflammation involved. So I think these are some of the ideas for the future. The big challenge is improving repair. We really don't have good ideas on how to do that. Stem cells for replacing cells that have become deficient or have become scarce in our bodies as we age. And then new technologies, new ways of maybe causing those stable cells that are damaged to have the good grace to die. And then we can use stem cells to replace them. So.
It's just an excellent and very elegant summary of it all. Thank you very much indeed. Julie.
So first of all, I'd like to thank Aubrey and the organizers for asking me to speak. I think I'm really excited about this conference. I think that the SENS Foundation has a holistic approach to looking at longevity and all the way from basic science to translational research, which is very exciting. So I'm going to focus on the translational piece of it. I'm the director of translational research at Wake Forest Institute for Regenerative Medicine. And I've been there for about 2 years. I've been in academia, industry, so a lot of different areas looking at all of the pieces of translation. And so this just gave me a great opportunity to put all that experience together. And so I always like to start out with our mission that we improve patients' lives through developing regenerative medicine, which is what drives us all at the Institute. And there's this force that makes us all move in the same direction because of this mission. So it's very exciting. We have over 300 faculty and staff, and that's led by our director, Tony Atala, which the Institute was established about 10 years ago, came over with about 20 research scientists from Harvard, and has grown significantly since that time. And I think everybody knows the team most for the bladder that was developed in a culture dish and then transplanted into spina bifida patients. So there's been a lot of firsts with Wake Forest since its inception, a lot of building constructs, miniaturized livers, corporal tissue, urethras transplanted into patients, and blood vessels in preclinical models. So there's been a lot of experience. We have over 30 different organs and tissues that we've worked on, about 150 different cell types, and a lot of different biomaterials. So there's a lot of disciplines that come together to be able to make up the Institute. When we look at the state of regenerative medicine, ARM kind of summarized that up a little bit with there's about 700 different companies in the space of regenerative medicine right now, all the way from big pharma to small privately held companies. And what I see, my viewpoint might be a little bit differently, but what I see is that years and years ago, I've been in the field for 25 years. I started out in bone marrow transplant back in 1989. And when regenerative medicine, when the term started becoming used, people really thought that big pharma would be a barrier to regenerative medicine. But what's kind of interesting is that if you look at all of the large pharma, they all either have a section of regenerative medicine or at least thinking about it, which is really a validation, which is what has been done in regenerative medicine all along. And so there was always that fear, but I think that they've jumped in, you know, even though that might change their dynamics and their strategy. But I feel like, you know, they're pretty much on board and have validated what we've been doing. And so these are the 4 different buckets that I really see in regenerative medicine and that I deal with at the Institute and the team of cell therapy, which is a very mature, in a very mature state, which came from the bone marrow transplant back in the 1950s. The tissue engineering being a little bit newer, and some of those that have made it to clinical trials or to the market, such as Apligraph or Dermagraph, where you actually have a scaffold and cells. Biomaterials, we look at biomaterials as well, and medical devices. So what are some of the hurdles? I think we've already heard of some of the hurdles as we've had full discussions yesterday, capital being a big one. When I listened to everybody talking about, well, the valley of death, I wish we could change that to the bridge to curing. I heard that a couple meetings ago, and so I thought that that was so great because it's very positive. And so hopefully we can change that, but getting capital to be able to commercialize the technologies is definitely a hurdle. The regulatory path is definitely significant and difficult, but I think when you're working closely with FDA, I think that there's a lot more, if you can educate them, it's a little bit more efficient. I think it's a lot more different than when I first started in the field back in the 90s, and I've been dealing with FDA since then. So I think it is a little bit easier, and I think that they understand and they want to get it to market, although there are a lot of hurdles, and I would like to see a different strategy in how we get regenerative medicine to the market. And then reimbursement, that's the big piece, because this isn't going to be sustainable without reimbursement. So what does our translational path look like at Wake Forest? We start out with proof of concept, important looking at farm tox, tumorigenicity as we move it to process and product development. We have a separate team that does that, move it in, and then as it gets to manufacturing, that's just a separate team that works on the GMP manufacturing of that to take it to clinical trials. And I'll just say some of the significant steps in translation. You want to be able to, you have a clinician that's very important on the team, so we need to be able to mirror what's done in the clinical trial at the preclinical stage. Otherwise, it's going to take a lot longer to be able to move it along. So early discussions with FDA become very important. And then just to kind of hit on the translational structure a little bit is that the translational research goes out to phase 2. Really at phase 3, beyond that is really different decisions, which might require a separate company or a joint venture or licensure. So you know, really when you get past phase 2, you have to make different decisions. And there's a lot of questions right now on when should industry get involved, when should academia release that, you know, and that's a big piece of it. And so I do think, though, that there's investors getting involved and looking at earlier on. I mean, of course, they want to de-risk it. They want the science de-risked, but I think that there is a lot more interest in looking at it early on as well. And then I do want to hit on the Japanese. I think somebody else talked about it, the fast track for approval, which is very exciting. And I think that's a really cool strategy where they're looking at, if they're seeing that something's efficacious with the first 10 patients and what we might call a phase 1, then they can take that to commercialization. They are able to take that for reimbursement to their national insurance. And so potentially you might be able to take something to the patient within 3 years, whereas it might take 6 to 7 years. Now, if they're not seeing efficacy or it's not, you know, definitely evident that it needs to move forward, then it might be hundreds of patients that they have to run through it. So I think this is a great strategy. It would be great to be able to look at that and somewhat help to model that in the US to speed things along. I know I was only given 5 or 10 minutes, so I wanted to just talk a little bit about, and I can just say high level, organ engineering. We definitely need to be able to replace as the population is getting older. And I'll just say at a high level, there's a lot of discussions either with decellularized organs or bioprinting organs. And there's a lot of pros and cons with both of those. The decellularized, you have the structure, the architecture, you have some of those growth factors left where you could seed potentially an autologous cell source. And when you look at the bioprinting, you have to be able to structure all of that. One of the significant issues is vasculature. You need to be within a millimeter of every vessel for the cells to be viable. So there's a lot of issues. So I'm not gonna go through it all because I don't want to steal the time from the last speaker. But there's a lot of challenges we're looking at. We want to look at an immunosuppression-free organ replacement. So hopefully we can use autologous cells. I think cell banking becomes very important in that effort. Hollow organs are much more difficult than complex, and the vascular structure as I described. So my thoughts are is that this field requires a multidisciplinary team, a holistic approach. We need to flatten. We have to have flat thinking, I'll say, which is to level out the silos. And the clinician is very important as we look at, as we're translating, we want to look at the final product to get their thoughts on what it's going to look like at the bedside to be able to mirror that. So thank you.
Julie, thanks very much indeed. And then to complete the journey towards commercialization, Howard.
Excuse me.
No, it's fine.
Thank you, Richard. And I'd like to thank Aubrey and the remaining organizers of the symposium for inviting me to speak today. I'm an endocrinologist by training with a particular focus in diabetes, and as such, I've devoted most of my academic and industrial career to diabetes and diabetic complications. Diabetes is a chronic and systemic disorder, and as such affects virtually every organ system in the body. Moreover, as I'm sure all of you are aware, it is an epidemic. In recent decades, both type 1 and type 2 diabetes have been on the increase. Now, the way I'm approaching the talk today is I'm going to take the current therapeutic strategies that are currently involved in the treatment of the disease, discuss their limitations, and then segue into a discussion as to how their an alternative treatment approach, that of cellular replacement, could offer certain advantages. Could I have the very first slide, please? Okay. There we go. Okay. Now, let's go back in time to the 1920s when Banting and Best discovered insulin. Here on the left, we have a picture of Banting and Best with one of their diabetic dogs. On the right, we have two of the very early patients who were treated with the insulin extracts. In 1923, you see before and after treatments. You see how they're emaciated prior to treatment and how they become healthy-looking individuals afterwards. Now, going forward, you can see we've had a number of technological advances over the past century. Notably, the development of NPH in the 1940s, that gave way to the check and finger stick glucose levels with various glucometers, and the beginning development of insulin pumps during the 60s. Recombinant human insulin was introduced in 1982, followed by the introduction of the first insulin analog, Humalog, in 1996. There have been many analogs approved since that time. More recently, continuous glucose monitoring has been implemented, and finally, artificial pancreatic development has been utilized. Artificial pancreas being combining the use of an insulin pump with that of continuous glucose monitoring. Now, where have all these technological advances taken us? Well, here are the results of the Diabetes Control and Complications trial, which was a landmark study involving 1,441 type 1 diabetic patients, completed in 1993. And we find that intensive glycemic therapy, here defined as the use of at least 3 insulin injections per day, resulted in a clinically significant and statistically significant reduction in the microvascular incidence of microvascular complications as well as macrovascular complications. So this was a, like I said, historic result, and for the first time proved that intensive glycemic therapy did occur. Well, is this enough though? Well, in spite of these impressive results, it did not eliminate the incidence of these complications entirely. Moreover, there was a price to pay, that for an increasing risk of severe hypoglycemia. As you can see in this slide, as you progress from the right to the left of the slide, as showing improving glycemic control as evidenced by a decrease in hemoglobin A1C, the risk for retinopathy and other microvascular complications decreased. But at the time, the risk for severe hypoglycemia, defined as requiring the assistance of a second individual, increased, such that those in the patients in the intensively treated group had a threefold increase in the incidence of severe hypoglycemia compared to the, those in the conventionally treated group. The implications of these findings is that there's an enormous impact upon the quality of life. In particular, it has an impact that goes beyond the patient, him or herself, in that it involves the caretaker as well, the concern about waking up in the middle of the night and being in the middle of a hypoglycemic episode. So, I'm here to discuss an alternative approach, that of cell replacement therapy. My company, ViaCyte, is developing a combination product known as VCO1, which is composed of two platforms, namely PEC-01, which is pancreatic endoderm cells, and Encaptra, an immunoprotective device. This device is implanted subcutaneously under the skin. The PEC-01 cells are precursor pancreatic cells and are designed to differentiate over the course of 2 to 3 months to form endocrine cells that produce insulin as well as other hormones. And it's designed to replace the islet function that is lost to the disease. I'm happy to announce that our IND was accepted by the FDA just this last week, and that we expect to launch the first in-human trial in the very near future. So, what are the current limitations of the standard of care therapy in this disease? Well, Type 1 patients have to do daily monitoring many times a day, sometimes as many as 15 to 20 times a day. In addition, they require multiple daily insulin injections or at least an insulin pump. They experience multiple hypoglycemic episodes. They have a diminished quality of life, as I've already alluded to. And lastly, there is the risk of long-term complications. We feel that there's vast potential with cell replacement therapy to either reduce or even totally eliminate many of these limitations. The cell replacement therapy is designed to mimic an endogenous endocrine pancreas. As you can see on this slide here, a human islet transplant appears histochemically very similar to that of a cell replacement therapy. One important note is that in terms of immunosuppression, islet cell transplants require chronic, lifelong immunosuppressive therapy. We feel that the cell replacement approach will not require this therapy. Moreover, islet cell transplants has very limited cell source, whereas we have an unlimited supply. Cost of therapy is very expensive. Cell replacement is much less so. And the surgical procedure of islet transplants is much more invasive. So, the VCO1 combination product is basically the cell therapy itself loaded into this encapsulated device that we refer to as Encaptra. This cell therapy source is a renewable source. It's derived from a cell line of embryonic stem cells known as Cy-T49, which was ethically derived under GMP-compliant conditions. We have multiple master and working cell banks that have passed rigorous infectious testing as well as other testing. We've reviewed it with regulatory agencies, and they find no restrictions on this particular cell line. It also has the capability of rapid expansion in that we can go to millions to billions within 2 weeks. Now, I'm preaching to the choir here because all of you know the vast potential of stem cell therapy. A single embryonic stem cell with proper cues can be propagated to pluripotent cells virtually indefinitely. Now, my company has been involved with working out a directed differentiation of these embryonic cell lines so that with proper cues, you can differentiate into the appropriate cell type. In our case, pancreatic cells, or more specifically, precursor pancreatic cells. This summarizes the manufacturing process. In approximately 4 weeks, we're able to go from human embryonic stem cells all the way up to the pancreatic endoderm. The first 15 days is involved with thawing the embryonic stem cells and propagating the cells, followed by a differentiation over the subsequent 12 weeks, at which point the PEC-01 cells undergoes extensive release testing and can be frozen, and they are stable up to 2 years. Now, what happens to the PEC-01 cells after you load them into the Encaptra device? Well, you start off with pancreatic precursors, which you can see over here on the left of the slide. Following the continued in vivo differentiation over the next 2 to 3 months, we arrive at the mature VC01 product, which constitutes 85 to 95% endocrine cells, which is composed of 40 to 50% insulin-positive staining cells, 20 to 25% glucagon staining cells, and 20 to 25% somatostatin staining cells. Moving on to the Encaptra drug delivery system, the materials of which it's made of are biocompatible and biostable. It retains the grafted cells, and very importantly to regulatory agencies, we have the potential after implantation of being able to remove the entire graft if any problems developed. In terms of efficacy, it does exclude host cells, so cells cannot move into the device, nor can they move out of the device. This gives us the immunoprotection whereby we can avoid the immunosuppressive therapy. However, oxygen, nutrients, and most importantly, proteins such as insulin and glucagon are freely transported, and this particular device promotes vascularization, which we see in this next slide. There's a robust vascularization process that begins as early as 4 weeks. The host tissue surrounding the VC01 undergoes the development of a thin fibrous capsule. And extensive vasculature begins to develop as early as 4 weeks. You see progression up to 8 weeks, and finally at 18 weeks. This shows you 16 weeks after the post-implant. On the left, you see an H&E staining of the graft. In the center, you see the cells, which represent the endocrine tissue that is derived from the PEC-01. The 2 gray areas immediately above and below this area represent the semi-permeable membrane, and you see how closely the blood vessels adhere to the outside of this membrane so that they're able to provide the oxygen and nutrients to the cells within the graft. And also here, again, the immunohistochemical staining, insulin in the blue, glucagon in the red, and somatostatin in the green. These represent the devices. I do have both types of devices with me if anybody wants to check with me after the presentation. This is the larger Encaptra 250 device. As you can see, it is freely flexible, we also are using smaller Sentinel devices, which we used in our rodent studies. The reason we're using the smaller device is so that we can implant multiple of these Sentinel devices and then explant them at time-specific, protocol-specific time intervals, and we can examine the histology and confirm that the same thing is happening in humans as occurred in the rodent studies. In terms of the implant locations, we expect it to be a minimally invasive procedure. Eventually, it could potentially be an outpatient procedure with only local anesthesia. The implant locations, at least for the purposes of the study, is to implant into the flank and lower back. The reason for this is we want to avoid the locations whereby the patients are using to inject that insulin. We do know that the devices are very robust against blunt trauma. We have simulated the impact of a baseball going 60 miles per hour with a 3 to 4-fold safety margin. However, that does not happen to be the case with the sharp needle of an insulin syringe. So we want to put these in a completely separate area. So just a little bit on the preclinical data that we have. This shows the use of this device to treat diabetic mice. You see on the left of the slide in red This is a normal, non-diabetic skid beige mouse. You see that the set point in mice is higher than that in humans. You're seeing the set point to be 150 to 160 milligrams per deciliter. At that point, you implant the VCO1 graft. As you can see, the red line tends to trend downward as the graft overrides the natural programming of the mouse beta cells. And so the set point is now reset to the human set point, which is less than 100. Going back to the left side of the slide, you see the impact of treating these patients with streptozotocin, which is toxic to beta cells. Hence, the animals become diabetic and become, frankly, hypoglycemic. However, once the VCO1 graft is implanted and allowed to vascularize and differentiate to maturity, you see the blood sugars come down rapidly under improved glycemic control. On the far right, we see the effect of removing the VCO1 graft. In the case of the normal non-diabetic animals, they reset to the mouse set point in the 150 to 180 range. In the case of those animals treated with streptozotocin, they become, frankly, hypoglycemic again. This next slide, which I just screwed up. There we go. Thank you. No. Okay, this is, I hit the wrong button, my apologies. Okay, this is the converse experiment, whereby we start off with non-diabetic animals and then we make them, frankly, diabetic and prevent, it's a primary prevention experiment. As you can see in the green and blue, these are implanted with empty devices, no PEC-01 cells, and they have the high mouse set point of around 150 to 180. In contrast, those who receive the loaded Encaptra devices, they have reset to the lower human set point, and these continue out for approximately 3 months, at which point half the animals are treated with streptozotocin. As you can see here, those animals with the empty devices become, frankly, diabetic and hence hypoglycemic. In contrast, those with the loaded VCO1 products, the diabetes is prevented and no hypoglycemic excursions occur. So, one last data slide. This shows the normal physiological regulation of this product. In the green, this is the animals without an implant. In contrast, when we use the VCO1 product at a vast overloaded dose that exceeds what we are going to be using in humans by about 25 to 30-fold, you see a much more rapid control of the elevated blood sugars following a glucose tolerance test and a return to a normal value. However, you do not see any overshoot when the blood sugars return to normal. Instead, they return to a normal level without any hypoglycemia occurring. So in our opinion, and in our experience to date, we have not been able to induce hypoglycemia with this product, which bodes well for safety concerns when we enter the humans. So that segues to the FIH study that we're getting ready to embark upon. This is going to be an open-label, dose-escalating study in diabetic patients with established but stable diabetes. We're going to be using both the larger therapeutic EN250 devices as well as the smaller Sentinel devices. The efficacy will be evaluated at 6 months, although we will continue the duration of treatment for up to 2 years to evaluate the duration and the durability of the biological effect. The endpoints that we will be looking at, to begin with, the earliest data that we get will be those from the explanted Sentinels and the histology that we examined there. This, we feel, will establish proof of mechanism since we will be looking at cell viability, the degree of vascularization, as well as the ability of the cells to differentiate into fully mature endocrine cells. Primary endpoints include safety and tolerability, as well as the measurement of stimulated C-peptide levels following a mixed meal tolerance test. We'll be looking at other secondary clinical measures, including the decrease in the exogenous insulin doses, as well as the frequency and severity of the hypoglycemic events. And hopefully we'll be able to report results in the not too distant future. And I'd like to conclude by thanking my colleagues at ViaCyte, as well as the generous financial support of several different organizations, notably the California Institute of Regenerative Medicine, as well as JDRF. Thank you.
Thank you very much. That's a fascinating story, and we've covered, I think, a pretty wide range of the translational issues here, from figuring out what's happening to senescent cells to actually replacing effectively by the device you've described. So where to sort of cut into this discussion? I mean, a general question I have, and maybe Judith will start on this, she laid out very elegantly the fact that you can either deal with damage and damage repair, or trigger apoptosis when something should have died, or you can basically replace the cells. Do we have a healthy balance between the investment in those two routes, or does that depend critically on the nature of the disease that we're looking at?
Well, you mean in terms of funding?
In terms of the whole research community.
Yeah. Well, I mean, this is a matter of opinion, but I would say right now there's so much excitement with regard to stem cells, and especially induced pluripotency, which eliminates some of the immunological problems, that there's been a movement towards much more funding and attention towards those types of therapies, which is not necessarily, I think, good, because in the end, if you put a very healthy stem cell into an old body, there's still going to be a problem. So we still have to find a way of dealing with that aging problem in order for stem cell therapies to be effective.
Is that a nice way of saying not enough on
that was a nice way of saying that.
The underlying biology.
So I think I'm interpreting your question as, is it more focused on proactive or reactive? And that's how I was interpreting it.
Damage repair versus actual just replacement.
Yeah, and so I mean, I feel like it's more on the replacement right now, because it's a pretty immature field, and so we're looking at how we can, as opposed to focusing on how we could fix the problem before it occurs. Yeah, so I pretty much think we're reactive and in the cell therapy field as well as the tissue engineering. Yeah.
Do you have a view?
Sure. One advantage of the replacement approach is that hopefully you would minimize some of the safety issues that you might otherwise have to deal with. You are simply taking a situation where there is an absent function and a depletion of a particular cell type, and you are simply replacing it with what's missing. And thus, you hope to achieve a more physiologically relevant therapeutic approach, and in that manner minimize potential toxic and adverse effects.
And obviously you've got this very cool combination between the cells and the encapsulating device. How will that actually be regulated? Have you figured that out with the FDA?
The FDA, I must say, has been remarkably enthusiastic. I hesitate to use that term. We're talking about a regulatory agency, but they really have been exceedingly supportive of our efforts. They have given us the opportunity for them to review certain of our protocols in real time. They have worked with us in discussing issues that they've had concern about, and we've provided them with answers. It was a very cooperative approach when we went through the IND review.
But they're viewing this as a medical device.
Well, CBER is actually driving the review, so it's regarded as a biologic.
Okay, because that's
with
interesting.
Consults with But they have consults from everybody else.
Right, because you're obviously going to get cells encapsulated, not just your company, but other
Exactly.
people will be encapsulating cells. But you think they're going to be regulated as biological therapeutics?
Well, I can't speak to the future as to what they may do because the FDA is the FDA and they always reserve the right to change their mind. But at least hopefully for the foreseeable future, since it is a cell product, they will regard it as a biologic.
So I'm sorry. Can I just ask, so they had CDRH involved as well as CBER?
Yes.
So it's a combination product, and then CBER leads the way. So typically when you have a cell product, you see that CBER takes that lead role. And
Exactly.
And I guess I'm also seeing the same thing when I'm dealing with FDA, is they're very cooperative. They want to see things move forward. I know we all talk about how difficult it is, the regulatory challenges, but I think that they try to do their best, and if we try to educate them, it's going to make it a lot more easier.
Judith, you want to add something?
I wanted to ask a question, which maybe from both of you. When you create a device, such as your device, have you done experiments to try to put those devices into old animals? Because, of course, the people who are going to need most of these therapies are going to be type 2 diabetics, right? They're not necessarily type 1. That's the major problem. And, of course, with age, there's all these comorbidities, so some of them undefined. So is there something about the age milieu that might interfere with the function of these tissues or devices?
Do you want to take it first, Julie, or do you want me to comment and then follow?
No, I see where you're going, and I agree with that. And on your first in man, whatever your population is going to be, you have to mimic that in your animal model. So it has to be that age group or age matched. But definitely you're going to have different issues as you look at different populations. So as you go through the clinical trial, you have to have represented that in your preclinical data.
In our development program, which is very specific, my comments are limited to our specific development program, I would have two comments. First of all, one thing that may not have been, I may not have emphasized sufficiently during my talk is that keep in mind that our product protects against both auto and allo rejection, but not xeno rejection. So consequently, all of our animal work has had to proceed in immunocompromised models. And these animals have a lot of comorbidities as a result. And as it turns out, the longest that we've been able to observe these animals is up to maybe 12 months or so, give or take, by which time they're dying from all of the other inherent mortalities and morbidities associated with these animal models, such as lymphomas and the like. However, in spite of that, that glycemic control was still excellent at the time that they died. Now, the second comment I wanted to make is that, remember I said there was a 2 to 3 month lag phase before these grafts would kick in with their biological function. And this is important to note because the devices have to become vascularized, otherwise the cells are going to die. Well, you have to think about that in terms of your target population, such as you are talking about. Diabetic patients tend to have vasculopathies, microvascular disease. If you try to treat an individual who has severely advanced complications, this device may not work. And that, I think, addresses your point of what about the aging population. So it's difficult to say. Now, keep in mind that type 1s tend to be more of or at least the onset of the disease is a younger population.
Yeah.
So if we could potentially treat these patients earlier, maybe we can eliminate a lot of the natural history of the disease that you commonly see with the development of all of these other complications, and it wouldn't be an issue. Sometimes when you develop a therapeutic product, you have to target an earlier stage population because there's this point of metabolic no return. Once you have had so much scarring, you just can't treat these individuals. You may be able to arrest further progression, but you can't reverse.
I just have one general question. I'm delighted that we begin to get some questions from the floor, but one general question just to bounce around between you, right? So you mentioned, I think, Julie, that there are 700 companies doing stem cell work, some big pharma companies along with those, and also institutes. Many of the problems would appear to be rather general problems, not just the regulatory interface, but the standards for measuring, cell content, cell type, cell diversity. Are there enough mechanisms in place to facilitate this dialogue across the industry and with the regulator, so that we don't have 700 completely different experiments?
Right, yeah, no, I think that's definitely a challenge, and I think people are working on that. FDA recently had a meeting trying to bring in all the folks that are involved in regenerative medicine to be able to discuss standards, how we can all work together, and there's a lot of folks looking at the standards, NIST and a lot of others, AABB, and FACT, although it was derived from cell therapy, also applies to regenerative medicine. So I think that there's a lot of work to be done yet to get everybody on the same playing field, and I think it's going to help, it's going to benefit everybody, because you really can't learn in a silo.
Yeah.
You have to be able to work with others to understand the issues, and then hopefully we can come to solutions together. Yeah, definitely.
Any points to add before we go to the questions? Please.
Dr. Foyt, if you already clarified this, I apologize, but type 1 diabetes is an autoimmune disease with
Correct.
autoantibodies destroying the islet cells of the pancreas. Have you already done things to make sure that the autoantibodies won't destroy these new pancreatic cells?
Well, you have to remember how the question is, type 1 diabetes is a autoimmune disease, and how can we protect the PECO1 cells from being destroyed in the process? And you have to remember that the Encaptra device into which the PECO1 cells are loaded do not permit either egress or ingress of cells. So that as it works out, yes, antibodies are small enough that they can get in and out of the device, so that the body knows that, hey, there's some foreign cells here and we need to do something about it. Unfortunately, you need other cell types, such as macrophages, to be able to get in to destroy them, and the membrane prevents the macrophages from getting within the graft itself. So hopefully that will solve that problem.
Again, Howard, a question for you. Congratulations on the IND. That's a long time coming.
Thank you.
Stephen Minger from GE Healthcare. Two questions. One is, on an islet by islet comparison, how much insulin do your differentiated ESL islets make upon full maturation compared to a normal human islet?
The best way to answer that question is in terms of the way they've done it in the islet transplant field,
Yep.
which is islet cell equivalence.
Yep.
And you basically need approximately 200,000 islets, IEQs, to become insulin independent.
Okay.
And so our larger 250 device is roughly 40 to 45,000 IEQs per device. And so we calculate, for the purposes of this initial study, that it will require roughly somewhere in the vicinity of 4 to 8 of the EN250s to make the patient become insulin independent. Now, keep in mind that 200,000 IEQ estimate is based upon 20% of the non-diabetic pancreatic capacity.
Okay. And the second question, and it comes out of the islet transplant programs as well, we know that almost all the patients who got islet transplants, albeit the marginal number of cells that they got, they all went back on insulin because there's turnover of the beta cells within the islets. Have you anticipated how often you may have to go back and re-administer your devices to particularly type 1 patients?
Good question. Unfortunately, I cannot give you a definitive answer. We've done experiments where we load different quantity of cells within the device. And it almost counterintuitively, as it turns out, it's not based upon the number of cells you load, but the volume capacity of the devices. And the reason we say that is that if you load in, say, 50,000 cells versus 100,000 versus 200,000, and then you look at the C-peptide response, the plateau is exactly identical for all three experiments. The only difference is the lag phase as to how long it takes to get to that plateau. If you load fewer cells, it takes you longer to get there. If you load more cells, you get there more rapidly, which means that the cells are capable of proliferating within the Encaptra device and self-propagating themselves up to a point. The reason your question becomes relevant is that if there was an infinite amount of space, we feel that they would proliferate indefinitely and you would not require retreatment. However, the reality is that you have a finite volume there. And so, again, it gets back to this gentleman's question over here. As you have beta cells don't last forever. They're going to senesce and turn over and die. And so you're going to have this buildup of cellular debris within the device. As that cellular debris builds up, you're not going to be able to proliferate and create additional beta cells. So we feel that the device is going to have a defined lifespan as far as its capabilities. Consequently, the answer is yes, we will have to replace the devices at certain points. At this point, as I mentioned to Julie, the longest we've gone out to is approximately 12 months because the animals are dying. That's one reason we've designed the FIH study to go out to 2 years so that we can see if they last. You know, at this point, we assume that based upon the preclinical data, they will last at least 12 months. But when you go from animals to humans, all bets are off. But,
Thanks.
I enjoyed the entire panel, but I have another question for Dr. Foyt. I'm going to give you some full disclosure. I'm Adam Leong. I'm a senior analyst for BioWatch News, and we're an independent source for public and private investors. That said,
thank you for the disclosure.
Yeah, I'm sure you're put to it, and so it goes both ways. But this is really a clinical science question. You know, I really want you. I appreciate that you present something on the what happened on the local histology when you implanted the device. I want you, if you could, provide some more color. And this really may get into the permeability of the device itself. So you have these cells coming in within the device, and right, they don't network directly with the rest of with the surrounding tissue, but it could be secreting growth factors in the surrounding area. Could you provide some color or even speculations about how it affected the surrounding tissue or even accelerated the implantation or integration, and whether you saw any differences or changes in some of the tissue-related biomarkers? I suspect your group probably had concerns about the downsides of the implant and how those. Love to also hear how those were addressed. And thank you very much for taking my question.
Very good question. In point of fact, we hypothesize that certain growth factors are secreted, although we don't really have the resources to examine that question closely. The reason we feel that there has to be certain growth factors involved is that we did do some experiments whereby we implanted empty devices, and unlike the loaded devices where you have this very well organized fibrous capsule form around the membrane, in the case of the empty devices, you get amorphous connective tissue forming and everything else. So obviously, the presence of the cells within the device are critical for this integration and organization of the fibrous capsule, as well as the ability to draw the developing blood vessels in to surround the graft. Sure.
Hi, this is directed to Dr. Allickson. So I'm trained as a regulatory attorney, and I wanted to ask about the role of the regulatory attorney in engaging the FDA in an operation such as what WFIRM has. Number one, are they being put to use as kind of an envoy in your experience professionally? Two, if so, how?
Okay, repeat the question again, the last half.
So basically are regulatory attorneys being put to use by organizations such as WFIRM in engaging the FDA, as you had described in your presentation? If so, how are they being employed?
Yeah, I think it's a great idea to have a regulatory attorney.
Okay, well, I'm right here.
Yeah. So we don't have our attorney involved in those discussions. I'm actually the head responsible for the discussions with the FDA and bring everybody else involved. But I think it's a great idea. I don't know if I'd be curious to know if the pharmaceutical world via site has that, but I think it would definitely be beneficial to have that voice.
What would be some of the things that you would expect a regulatory attorney to bring?
Well, I mean, I think that an attorney, just by their very nature, are great negotiators, and they can present kind of the arguments and the reasonings why a doctor or a researcher needs to get certain things done, and using kind of, I mean, I'm not really well prepared to make that sell for myself right now, but just generally knowing from my experience how I help people in my work, I seem to be able to unlock doors and pave paths to make it easier to have those discussions between Silver Spring, where the FDA is, and the scientists themselves. Mm-hmm. So I think that's something that I would love to see more of, more regulatory attorneys kind of at the front end as opposed to at the back end whenever you're dealing with litigation.
Yeah, and I think, it's a whole multidisciplinary team that comes into those meetings actually, and all the scientists that are involved in the research too, because we want them to be able to get their questions answered as well. Although there's discussions on how, what we should and shouldn't say as we go into these meetings. So it's important because you only have an hour with the FDA generally on a teleconference, and so you want to make it effective and productive. Although you do have ongoing discussions after that as well. But I think it definitely could be helpful to have a legal regulatory voice within the team.
It's been
sorry, please. No.
It's been my experience that there are regulatory courses and various levels of certification that you can do in terms of companies. As Julie pointed out, I think there's a limit to how much the trainee can do, because both in terms of resources, plus you only have limited exposure to various regulatory agencies. But I have seen situations where there have been extended internships associated with companies whereby trainees can come in for the experience.
Thanks.
Yes, hi, Dave LaRocca from Biotime. So there's these experiments where they're called heterochronic parabiosis, where you hook a young mouse to an old mouse and they share circulatory system, and soluble factors in the circulation of the young mouse can restore some of the regenerative ability within the older mouse's stem cells. And I'm wondering if the panel could comment on the role of the soluble factors in when you transplant, say, young cells into an older person or animal, and whether encapsulated or as a tissue.
Actually, it works both ways. So there's something about the young systemic milieu that will improve regeneration in an older animal, but there's also something that the older animal produces that suppresses regeneration in the younger animal. So I think this is where it's going to be really important to get at these basic aging processes, because it's going to affect how anything that you do to an older animal is going to improve the health. We have to be able to understand what that milieu is and how to correct it to make it more youthful.
I feel the plot of a science fiction film coming on at this point.
After these papers were published about these parabiosis experiments, there's been this run on virgins and right, virgin blood. It's like, but of course we don't have many in Berkeley, so we weren't worried.
You don't have many in Berkeley. That's a big surprise.
So this is Shane Loring from the Scripps Research Institute, and what I'm curious about is the way that the FDA dealt with using pluripotent stem cells as opposed to cells that have much more limited potential? Because I know one of their concerns is that you may make something that will actually be harmful. The cells will, since you're putting the cells in before they're fully differentiated, how did you convince them that those cells were only going to be making islet cells and not something completely abnormal that might be dangerous to the patient?
Well, in our case, during the manufacturing process, we monitor certain biomarkers such as OCT4, which is
I wasn't thinking about teratomas. I was thinking about differentiation to another cell type.
Oh, you mean like after implantation?
Yeah.
Well, we've demonstrated through animal and cadaver studies that we can use ultrasound to monitor the injection site. And we will be using that technology during the confines of the study, as well as in a follow-up period following the termination of the study.
So was that reassuring to the FDA that you were going to do those studies? Did they, I mean, I know in another instance this is exactly what they were thinking about. So I was wondering how you ameliorated their concerns about cells differentiating perhaps too well or into something else.
Well,
but apparently
I think we're almost more concerned about the possibility than they are.
No,
quite
exactly,
frankly.
and that's a good position to be in.
Because, I mean, in one of our teleconferences, they made it quite clear that their major, in their minds, their major safety concern was for hypoglycemia.
Yeah.
And in fact, they did make a comment that, well, certainly there are certain, quote, other theoretical safety concerns,
right,
such as teratomas, unquote.
Yeah.
So based upon that comment, it doesn't appear to be high on their priorities.
That's good. I hate to see them freak out about things they shouldn't worry about.
They really are impressed by the ability, our ability to be able to explant the graft.
Yeah. Yes.
If we get into trouble for whatever reason, whether it's acute rejection, an inflammatory response, a teratoma, or anything else.
Right.
Now, if the device does become inadvertently punctured, yes, the cells could spill out into the patient, but based upon experiments that we have done in rodent as well as baboon, the cells become basically destroyed in a punctured device within a 2 to 3 week period.
Oh, yeah. Okay, good. Thank you.
Hey, my name is Ruby Yan Rusai. I'm with Applied Stem Cell. My question is for Howard also. So I'm just thinking about the lifespan of the device after you implant. So you mentioned in animals it took about 2 months for the cells to differentiate into pancreatic specific cells. And then you can still see effects like over to a year. So it's like 8 months period when the device is functional, right? So then for the humans, I guess we don't really know how long it's going to take for the cells to differentiate into the pancreatic lineage cells. And then we also don't know how long the effect is going to last because the cells are going to die off. And so that's the question about lifespan. So if you can get 6 months out of it, I think it's a very big improvement for what people have to do right now, right? And so I just wondering if you thought about doing a few things to actually to make either differentiation process more efficient, so you shorten the time of the cells from embryonic stem cells to pancreatic cells. And so that would extend the lifespan of the device.
All good questions. First and foremost, from a patient viewpoint, the lifespan is probably not a concern. You know, many patients have come to us and they say 2 years is, you know, an unbelievable home run. 1 year is fantastic. 6 months, give it to us. We don't care. And so from that perspective, you know, no concern. From a commercial viewpoint, obviously there are implications and we want to prolong the lifespan as much as possible. Now, keep in mind that there other organizations have attempted a similar approach with mature beta cells. And these experiments have largely failed. And the reason is that the beta cell is probably one of the more active metabolic cells in the body. And so consequently, you put these cells inside an encapsulated device, and they don't have the oxygen and nutrients to support their continued existence, and they die. That's one of the advantages of our approach. We're taking precursor pancreatic cells that need 2 or 3 months to continue their differentiation to the mature beta cells, but you need that 2 or 3 months for the blood vasculature to develop to support their continued existence. Now, you might say, along the lines of the question that you asked, maybe we could differentiate them a little bit further, but not all the way to the beta cell. And that's one approach we have considered as a backup plan, but right now our current approach seems to be working, and until we actually get it into man, there are so many unanswered questions, we may just end up chasing our tail. So our approach was to, let's go ahead and get into man, see what the time course of the vascularization and the differentiation happens to be, and what the functioning life of the graft happens to be.
Thanks for the great questions, and I think it's been wonderful to go through this case example, and you're at a wonderfully interesting point, which we'll watch very closely. Any final comments from the other members of the panel in terms of, how this field should go forward?
Well, we're living in such interesting times. That's a curse, by the way, in certain cultures. But I think it's a blessing. It really is. I think this is great. We're going to revolutionize medicine, I think, in the next decade.
I think this is the way that we start with these conferences, like SUNS has put together, getting the researchers, the VCs, the capital, regulations, scientists, clinicians, everybody together to think of how we can move this field forward efficiently.
So a big vote for hope and a big vote for convergence.
Yeah.
Let me invite you to thank our speakers.